Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay, we shall start today's class. In our today's video, we shall discuss about artificial hybridization. In our previous video, we have discussed about pollen pistil interaction, about this structure. Okay, we have discussed about this structure in detail. Today we shall discuss about artificial hybridization. Now, what is this artificial hybridization? Artificial hybridization, it is a process of transferring desired pollen grains onto the stigma by preventing contamination from unwanted pollen to obtain improved crop varieties. So this is what it is about artificial hybridization. Now what does it mean? We shall know about that. Okay, this is the definition for artificial hybridization. Now it means that it is a process of transferring desired pollens. Okay, desired pollen grains are transferred onto the stigma. The pollen grains which we want, which we desire, those pollen grains are transferred to the stigma by preventing contamination from unwanted pollen grains. Okay, so we shall take one of the plant. This is one of the flower. This is another plant and another flower. Okay, so we should imagine these are the mango plants or mango trees. Fine, mango trees, or you can think about a rose plant or some other, whichever you want. The hybrid varieties of the plant which we are getting, that is by using this artificial hybridization. Now this plant, as we are taking an example of mango, this plant will produce small sized and good in taste mangoes. Okay, now this plant will produce large size or not that good taste. Okay, better taste of mangoes. Okay, now these are the two different plants which will produce, both will produce mangoes. Now one will produce a small size but taste is very good, it is very sweet, whereas the other plant will produce large size mangoes and it is not that good, it is quite better. Okay, it is not sweeter. So what do you want? We want what? Large size and good in taste or sweet in taste. Okay, or a tasty fruits. So we want that one. Now we wanted to hybridize these two plants. So what we shall do? We shall proceed with the artificial hybridization. Now from this plant, what pollen grains are there? Okay, the pollen grains are collected from one of the plant, either this plant or that plant, whichever plant. From one plant we are collecting the pollen grains. Now these pollen grains will be dusted on the stigma of the another plant. Okay, the pollen grains which have been collected from one plant is dusted on the stigma of the another plant. Now that pollen grain will germinate and it will fertilize and it will produce fruits and seeds. Now the fruit which is produced here, it will be having large size and good taste. Okay, the combination of both. Okay, large size from here and taste is from here, because pollen grains have been taken and dusted over stigma on this, and what fruits and what seeds are produced by this, those will be having this features. Hope you understood this. Okay, even as I'm giving the example of rose plant. If you imagine about rose plant, this is a red flower, this is a white flower. If you wanted to get a in between like a pink flower, then you are seeing you will take from one of the plant pollen grains, you will collect and dust it on the stigma of the another plant, and you will allow it to germinate, so that pollen grains will germinate and fertilize, and what next plant will be produced, that will be having pink color flowers. Okay, this is how hybridization method takes place. That for that we call it as artificial hybridization, and this is the definition of it what I gave earlier. Now this is how in ordinary terms I have given an examples. Artificial hybridization is achieved by two of the techniques. First one is emasculation, and second technique is bagging. Okay, the first technique is emasculation and second technique is bagging. These two steps will go through in artificial hybridization. Emasculation: if a flower is bisexual. Bisexual means the flower which consists of both the sex organs. Is it? Both the sex organs, females and a male. If both the sex organ is present in one flower, like one flower, it do have pistil and anther. When it consists of both, then we call it as bisexual flower. In this kind of flower, emasculation is done. What is that emasculation? The technique of removal of anthers from a bisexual floral bud. Okay, now from this plant, if I show this plant here, this is a flower. It consists of anthers and a stigma. Both the organs are present. Now when this flower is in the bud stage, if it is in the bud stage, still it has not yet bloomed, in that stage only anthers have been cut down. Okay, with the help of forceps or scissors, you can cut this anthers from a flower. This is a flower, these are the anthers. Okay, you are cutting this anthers. Why we are cutting this anthers? Because if this floral bud grows into a flower, then this anther matures and there are chances of containing pollen grains. It will produce a pollen grains. Now there are chances of self-pollination, but we don't want self-pollination, we want this kind of pollination. Okay, so as if we are talking about this flower consists of both male and a female part. Stigma is there, pistil is there, and there is presence of androecium also. So when both are present, then there are chances of self-pollination. So when this flower is in the bud stage, in that stage only the anthers are cut down. Okay, anthers are removed with the help of scissors or forceps, and it is allowed to grow. Okay, now what we are doing, only stigma has been left, as we found stigma is present here. Okay, like that. That process we call it as emasculation. Okay, the technique of removal of anthers from bisexual floral bud before their dehiscence using a forceps or scissors is called as emasculation. Next one is bagging. Now when the flower is emasculated flower means from those flower from which anthers have been removed with the help of scissors, that flower we call it as emasculated flower. Now that flower is covered. Okay, emasculated flower is covered with a bag made up of butter paper. Okay, now this flower whose anthers have been removed, emasculated flower, it consists of only stigma, that is pistil, right? Now this flower is covered by butter paper, that is in a bud stage only. Butter paper, it has been tied. Okay, so with the help of butter paper it has been covered. Why it will be covered? So that other unwanted pollen grains could not reach here. Is it? Natural pollination takes place, but we don't want that natural pollination to take place. We want our desired pollen grains to reach here on the stigma. So naturally pollination takes place, it should be avoided that. So for that reason we are covering this flower with the butter paper. Okay, if you find here, now this we said it carries stigma, we said pollination takes place here. In this anthers will be removed, okay, with the help of emasculation, and after removal then this flower will be covered by the butter paper. That process we call it as bagging. Okay, butter paper, when this flower has been matured with a stigma, then what we'll do, we'll remove this butter paper, the cover which has been covered that flower, we will remove this butter paper, and from the another flower, the desired flower, from that we shall collect the desired pollen and we will dust it on it here. Okay, if we are not covering the unwanted pollen grains naturally from some other plant, it can reach and get fertilized. So for that reason we are not allowing this. So for that reason we are bagging, or we are covering this flower bud. Okay, so that it after getting mature, after blooming the flower, we are making it to get pollinated. Okay, so this is how we are doing bagging. Okay, now after dusting the pollen, now we have dusted the pollen grains here of the desired pollen. Then after dusting the pollen also we will rebag. Okay, In a bagging, we have rebagging also. Okay, the mature pollen grains collected from the anther are dusted on the stigma. Then the flowers are rebagged and the fruits are allowed to develop. Okay, now we'll after dusting the pollen grains, again we'll rebag so that it should not undergo any kind of contamination, and will allow that one to develop into fruits. Okay, so this technique will be followed, that we call it as rebagging in a bagging. Fine. So I have talked about bisexual flower. If bisexual flower is there, emasculation takes place and a bagging and a rebagging. If there is a unisexual flower, if we are talking about unisexual flower, in unisexual flower what happens? Male flower is different, female flower is different, right? When these two flowers are different, in that one step is eliminated. Which step? Emasculation. In a bisexual flower, because it consists of both the sex organs, male and female, so emasculation is necessary. We have to remove the anthers. Because it is a unisexual flower, no need of bagging. In a unisexual flower, emasculation. In a unisexual flower, there is no need of emasculation, only it undergoes bagging and a rebagging, as we have discussed earlier. Okay, once again, if I am showing it here, this is one plant, this is another plant with the desired characters. Now this plant which has produced flowers, in its bud stage only we are removing its anthers. Its anthers are removed. Now that step, what we call it as removal of anthers with the help of scissors, is called as emasculation in a bud stage. As soon as emasculation has been done, what will be left? Only stigma will be left. Okay, pistil will be left inside that. So for this flower, what we'll do, we'll cover with the butter paper. Okay, now covering of the flower bud, emasculated flower with the butter paper, we call it as bagging. That step we call it as bagging. So as this bud has been mature, then we'll remove this butter paper and dust the pollen grains from the desired plant on the stigma of this flower. After dusting the pollen grain, what happens? Again we are proceeding with the rebagging. Again we'll take a butter paper and cover it with this flower, and after some time we'll allow it to develop into fruit. Okay, so this is what about hybridization, artificial hybridization with the emasculation and bagging and rebagging. In a bisexual flower, emasculation and bagging takes place. In a unisexual flower, only bagging and rebagging takes place. There is no step called as emasculation in a unisexual flower. About this information you have in your textbook on page number 33. As you can find here, it is here about artificial hybridization. It is useful in crop improvement program to improve the crops. So all that information you have in these paragraphs. Okay, so you have got the information about this. If you have any doubts, any clarifications, if you want, you can comment it in the comment box. I shall meet in your next video. Stay safe, stay healthy.